Video tutorial we will be discussing about T cell maturation. So T cell is originated from hematopoietic stem cell, which is differentiated to lymphoid progenitor, and from the lymphoid progenitor the T cell maturation takes place. So the hematopoietic stem cell primarily present in lymph in the embryonic stage, and in the adult it is transformed to bone marrow. Okay, and the for maturation of T cell it has to go to the thymus so that it can be mature. So what happens in the bone marrow, the T cell precursor are present. The thymus release some sorts of chemical like thymosin, thymotaxin, thymoprotein, which induce the bone marrow to release these T cell precursor to the blood vessel, and the thymus have blood vessel at its middle portion, which is known as corticomedullary junction. The thymus has upper portion known as cortex and lower portion known as medulla, and in between this layer is known as corticomedullary junction, in which the blood vessels are passed as endothelial vesicle. So these T cells are released from bone marrow to the blood vessel, and they form endothelial venule so that it can pass to cortical region of thymus. So what happens? From bone marrow, these T cell precursor goes to thymus to blood vessel by endothelial venules to the cortical region. What happens in cortical region? There is for T cell maturation there is two things takes place: positive selection and negative selection. We will be first talking about positive selection. The positive selection occurs at cortical region. So this T cell precursor from the endothelial venule first go to cortical region. In the cortical region there is a cell known as CTEC, cortical thymic epithelial cell, which have two types of MSC, both MSC1 and MSC2. MSC stands for mature histocompatibility complex. So let me tell you one thing. The MSC1 is present in all nucleated cell except RBC. As RBC has no nucleus, so it don't have any MSC. But MSC2 is present only in antigen presenting cell like B cell, macrophage and dendritic cell. So when this T cell precursor goes from venule to cortical region, it possesses two types of receptor, CD8 and CD4. So we can say this T cell as CD8 plus and CD4 plus. Okay, these CD8 and CD4 receptor have specificity. What type of specificity? The CD8 receptor bind to MSC1 and CD4 receptor bind to MSC2 only. So when such type of situation is arising, only one thing have to happen: either CD8 will bind to MSC1 or CD4 will bind to MSC2. So when CD8 bind to MSC1, it will give CD8 plus, and when this CD4 bind to MSC2, it will give CD4 plus cell. So what happened, the CD8 plus form T cytotoxic T cell in future, whereas CD4 plus form T helper cell in future. Okay, CD8 plus form cytotoxic T cell in future and CD4 plus form T helper cell in future. And this situation is totally dependent on chance factor. Either CD8 will bind to MH1 or CD4 will bind to MHC2. There is a 50% chance either this thing will happen or that thing will happen. And after that, there will be a conclusion that CD8 plus or CD4 plus cell line will be formed, and they will give rise to either cytotoxic T cell and T helper cell. Thank you. In the next video, we will be talking about negative selection.